start. So for me, it's a particularly great pleasure to have here as a distinguished speaker Professor Chandrajit Bajaj. He's a visualization chair, Computational and Applied Mathematics, Professor of Computer Science, Director of the Center for Computational Visualization, and I'm sure I forget many other titles, but personally his main title is also my advisor for the PhD way back in Purdue, and then actually we moved in Texas together when I was finishing my dissertation, and he was actually starting his new large center at the University of Texas at Austin. So of course he's well known, he does not need a huge introduction. All this work in visualization, analysis of data, and more recently in the area more of computational biology. So thank you, it's great to have you here.
Thank you. Thank you, Valerio, and I should also thank a lot of friends that I have in the University of Utah. Some of them are here in the audience. Also thank you, Chris, for the kind invitation. It's always good to come to a place where you see success in its many forms, and also to see how well Valerio is doing, because after all your academic children are part of your life. In some sense, it's also a good time, because Valerio left about 10 or 11 years ago, and right after he finished his PhD, I figured that we had worked on this area for a while, and like every computer scientist, you don't like to stay on a problem for too many years. And so I thought I'd embark on a new field, possibly where some of the tools that I'd acquired could be applied to. So I took a sabbatical about a decade ago, and I'm just now finishing a couple of books. And when you try to finish books, you feel that you've done enough in this area. But as I'll tell you in this talk, there's a whole lot of problems I haven't been able to solve. And there are still numerous challenges that several people who are younger than I am, and as well as more energetic, I'd invite you to jump in, because this area is ripe for the computer scientists in us, as well as the mathematician in us. So this area of, in some sense, molecular modeling, computational structural biology, and so on, has two aspects, and that's why I kind of broke this talk up into two little parts, but as I'll tell you at the very end, all these parts kind of fit together to work out the puzzle of the science or the discovery that we are after. I'd like to start off by acknowledging several people, because success is always gauged, but has many fathers and mothers and so on. My group consists of a mix of people from computer science, as well as mathematics, as well as people from biomedical engineering. But I owe a lot of thanks to, and some of their work I will mention in this talk. But I owe a lot of thanks to my senior collaborators, without whom I would not have learned the field, and I'm still continuously talking to them. And in particular, I'll mention people like Tim Baker, who's a microscopist, people like Wachu and Steve Lutke, who are also microscopists, and Joachim Frank. Then people like Andy McCammon and Mike Holst. Andy McCammon is a biophysicist who I work often with, and people like Art Olson and Michel Sana, who are biochemists as well as also in the field of visualization. And so you'll see their names crop up ever so often in this talk. But perhaps the biggest inspiration of all of this comes from a person who delivered this wonderful lecture, a person who's known to many of you, Richard Feynman, in 1959, where he delivered this lecture of "There's plenty of room at the bottom." And not only was that a visionary lecture, which has so many statements that are coming true today, but one particular one is he mentions the problems of chemistry and biology can be greatly helped with our ability to see what we are doing and to do things on an atomic level is ultimately developed, a development which I think cannot be avoided. And part of this exercise is that some of this is coming to come into fruition. And it's, so again, a fruition that has been achieved by a mix of biophysicists, biochemists, microscopists, computer scientists, and mathematicians. And it's part of this interdisciplinary work, which is so active also here, that I like to report to you. So let me start off by telling you a little bit about what the electron microscope does. It's like the visual eyes to the nano world. Unfortunately, or I should say first fortunately, it can give you images down to the nanometers. Right? But unfortunately, it doesn't have a temporal resolution. So you don't have a video camera. So you can't see things in motion, but you can see things in fine granularity. And that's the imaging that you need to help you visualize structures which are at the, what I'd call subatomic level. But using computational techniques, we can actually, and using super sampling, we can actually go and beat that resolution, go down and start to see things at an atomic level. And that's become one of the true remarkable revolutions in this last decade only. One particular thing is called single particle cryo-EM, and I'll tell you a little bit more about this. But you can see images of viruses. These are structures, say, about 100 nanometers in diameter to about 200 nanometers in diameter. Some of these viruses are enveloped. And these enveloped viruses, like HIV is an example I'll show you a little bit later on, but the influenza virus was an envelope virus. And this envelope is actually garnered from the same cell, so it's made up of bilipids, the same material that makes up the boundaries of all our cells. And it's like a cloak that it uses to gain entry, as well as a cloak it takes away from it when it buns out from a certain cell. This is a figure of tobacco mosaic virus, which is helical in structure. So in looking at all of this, what computational scientists have been doing is trying to go back and piece together, what the missing dimensions are. And clearly time is a missing dimension in this static snapshots that we have. And so this is just a scientific animation, which is partly interpretive and partly, of course, from our own imagination of how something like a bacteriophage, a virus that infects bacteria. Viruses are parasitic, and so they have to take over a host cell. And their mechanisms of gaining entry into these host cells is something that is important to learn, because that is one of the basis of what we call, if you're trying to come up with a cure, we need to go back and understand the life cycle of this virus. So the first set of challenges that I came across 10 years ago was the fact that people are wanting to understand, before they can even develop a cure for a certain disease, is to find the causative nature of the disease down to the molecular level, and perhaps down to the interaction level, which is sometimes at the atomistic end. Of course, in certain cases, there could be quantum level effects, but at this talk, I'm only going to start from the atomistic level up. So some of the challenges were key were, how do we go back and derive structure from images? So this example is that of a single particle cryo-electron microscopy. And this is akin to doing 3D photography, but using only a single image. So how do you recover 3 dimensions from one 2-dimensional image? Well, the little aha that people had about a decade ago was, we can get identical particles purified using robots and other purification methods. So this is 1 million copies of the same virus. And it's in vivo. It's in its nativity. So it's in its solvent bath, or I should say it's really in vitro when it's purified and removed. And in this in vitro bath, it's in its own Brownian motion. So if I had 1 million ballet dancers all dancing about in their own dance, and if I took a single snapshot, the probability is, of course, close to 1, that I'd get a fairly good sampling of various orientations from a single image. And so if I can grab those particles from this image, and I can guess their orientations, then I can do a reconstruction by traditional methods of reconstruction, and I'll tell you about one of those methods also. So these are then the computational techniques that needed to be solved before which we can do structural elucidation from imaging. And imaging now at the microscopy level, where we don't have the ability to take multiple snapshots and also snapshots fast enough that we can gauge their temporal activity. So the challenges are pretty much, or the computational techniques are very similar to most kinds of image processing that we have dealt with, perhaps at the macro level, but there are some newer challenges. And some of determining what works and what doesn't work and where new techniques have to come up is part of the exercise that we've been involved in, as well as several others have been involved in. One particular nasty thing is this problem called the limits of the Born approximation. So most of the imaging, like X-ray imaging, CT imaging, and so on, is giving you two-dimensional slices through the body, but there is a reconstruction. So what you're getting is the scattering image, or the scattering signal, or the transmission signal of an X-ray through a material. The material could be the human body. And when you collect that, and you get the scattering signal from different poses, then you can go back and reconstruct a three-dimensional volumetric map or an image of the domain. The fact that that two-dimensional image is considered a projection is because the X-rays for the material are partly absorbed, but they are in some sense incoherent, and they make their way kind of directly through. There's a little bit of scattering in the material, but that scattering can be ignored. And so hence we assume the two-dimensional image is a projection of the three-dimensional volume. And hence when you're thinking of it as a projection is linear and it's invertible, hence you can go back and invert and build the three-dimension. When you're starting to look at microscopy, and you're using, of course, now 1,000 times more powerful X-rays, these are in the million electron volts, and you're passing it through the material in some sense gets destroyed. It's of the ferocity of something like a Hiroshima bomb exploding on that object. So that particle gets destroyed, but by clever means you can capture a signal. Now the wavelength of your X-rays are about the same resolution as the features that you want to detect. And anytime you hear that, you say, oh, is the features that I'm detecting of the same wavelength as my X-rays, is the Born approximation, or this projection operator, valid anymore? And the answer is no, it's not. So hence the forward operation is no longer linear, and that poses a big challenge. But before we can solve the nonlinear forward operator and hence the reconstruction, we are still assuming how far can we get with approximations with assuming that it's a forward operator, which is a linear projection. And this is one of the key things that while we have efficiently been able to solve things for projection images, we have not yet even addressed nonlinear projections or corrections of reconstructions for that. So this entire, once you acquire the images, and by no means is acquiring the images a simple art. It requires a lot of sophistication because there can be a lot of things that go wrong in a microscope. The optics causes error, there's short noise, and the scattering signals, the way the data is collected by collecting photons and so on, leads a whole bunch of approximations. But once you acquire the images, the challenges of going back and building something like a three-dimensional reconstruction, and then finally finding those structures that you were seeing in that video. So starting from that single image, one can build a three-dimensional map, and from that map you can actually build a model. A model that you can now do various types of analysis with. And the analysis that we will be talking about is how you do drug design and discovery, how do you look at interactions between things. Now I'm not going to spend time on each of these details, but I thought I'd pick out one or two crucial problems and tell you the flavor of the techniques, and some of these you'll recognize and some of them you might not. But this problem of going back and doing a reconstruction, which is taking a single image plucking out each of the particles. And you can see the signal to noise ratio is pretty low. Hence there are challenges there. But with, with all the progress in image processing, you can go back and extract these. And then you have to use learning algorithms to go back and estimate the orientations. Unlike tomography, where you know the pose of the projection images, here you don't know the pose, so you have to estimate them in an iterative loop. And once you estimate these orientations, you can then go back and try to come up with a good, valid reconstruction, especially where the data is very noisy. So the traditional technique of reconstruction has a large history of several people who applied it to imaging, CT imaging, MRI, and others. And more recently, one has started to look at how this affects microscopy. And so there's still, research going on. There's several resource centers that have various packages. But this was one of the challenges that I started to work on fairly early on. And it's only recently that we got some handle on it. And this handle is something that still is work in progress, I should say. So this 3D EM reconstruction problem, we pose it as what we call a variational formulation. So what we are trying to do is, given a set of 2D images where we are considering as projections, and we know the projection directions, go back and reconstruct the three-dimensional function from which these projections are, of course, obtained. So what you normally do is you say you want to something like solve this as an optimization problem. You want to find that function, which is the unknown, which in some sense minimizes some norm on the data space. It could be least squares, it could be minimizing the sum of some other p norm or something like that. But as many people know, this formulation is highly ill-posed, because it's ill-posed especially for microscopy, because you cannot prove an existence of a true solution. And furthermore, the number of projection directions you get is still sparse. You don't get a complete uniform sampling of all of orientation space. So what in mathematics people have, of course, every time you have ill-posed problems or inverse problems, you look at what are called regularizations. So this regularization is acting as a term that you throw into your what's called your functional, which you're minimizing over, and it acts as a not only a penalty term but also as a guiding term towards coming up with a true solution. So such regularizers are like any information you have about the particle or the domain. You say, oh, the domain interfaces are smooth, or the domain has a certain volume, or the volume ranges are between this and this, are prior information that you have about the object that you're trying to reconstruct, and you fold those into various regularizers. And so typically, the choice of the regularizers becomes the key computational problem that you have to solve. Once you do that, of course, there's the numerical problem of how do you go back and solve this stably. So such challenges are still, I should say, a great avenue for future researchers to get into. While we made some progress, and we've been able to go back and come up with these regularizers that have been doing slightly better than what others have done, the problem is by no means solved. So one measure of success is, of course, in the reconstructed map that you have, what kind of features can you elucidate? So the resolution of the reconstruction that you talk about is not the pixel resolution of the volumetric data that you reconstruct, but it's really the feature resolution of what kind of domains and subdomains can you identify. So this is a reconstruction using this tomography, not tomography, but single particle electron microscopy of a macromolecule called Rho-EL, Rho-ES. It's a chaperonin that actually is very essential to the body in two purposes. One, proteins are created in the body, and every time a malformed protein is created, this molecule tries to actually shape it into the correct form. If it fails, of course, the body has other destructive processes that will destroy the protein into its amino acids, and then again, that protein has to be remanufactured. So the body has all of these controls. This is also a marker for cancer. So when the body has excess concentrations of chaperones, then you know that a whole bunch of proteins are being produced at a greater requirement than is on average or is necessary. And so these are the one of the indicators. So it's important to understand not only its structure, but from its structure, its function. And that's why this is a key molecule that we are not only interested in reconstructing what the overall topology and geometry is, but also the amount of features we can elucidate. So one of the features is that this consists of a symmetric collection of protein, which is called a D7 symmetry. And in this D7, twofold D7 symmetry, each little colored piece is a single protein. And a protein has, what we call a secondary structure. It folds its collection of atoms into helices and sheets. And that's shown here by these glyphs as little cylinders, which are helical cylinders, and sheets are shown as surfaces, which are purple colored. From this density, by segmentation techniques, which several of you here are very proficient at, one can extract this density. From this density, you can then also elucidate these helices, and you can extract these features. And in those cases where you have, alternate models of the protein, you can actually do one better than going and extracting these helices and sheets. So I should say that helices and sheets are groups of atoms which are what we can call at feature resolution of about 5 to 10 angstroms. Atomistic level detail is feature resolution of about 1 angstrom. Bonds are about between 1 and 2 angstroms. An angstrom being 10 to the minus 10 of a meter. So when you can extract models which are 5 to 10 angstroms, you're 5 to 10x worse than what you need to be for understanding how proteins interact with each other. But by crystallography means, sometimes you can actually purify a protein, put it into a crystal, use X-ray diffraction, and get atomistic-like coordinates. It's also approximate, but gives you another data set. And that data set can be then fitted. And this also is done computationally. It's not a mechanical fitting, but it's a computational correlation fitting. So such problems of structural elucidation are all important to not only go back and reconstruct the model, but then actually go back and build a best resolution quasi-atomic model that you can to do further processing with, which I'll also tell you a little bit about. So just so that I'm not giving you just a high-level view, I thought I'd give you details about one of the secondary structural elucidation algorithms. And also, it's an area that, since Valerio was here and was acting as my host, I thought I should mention he has done a lot of work on as well. And it's a technique on, in substance, computing what are called stable and unstable manifolds. And so this is a theory that has been developed by several people. And one of the nice things was, can we actually harness it for real data and actually come up with something which is meaningful? So in the times past, when I was looking at this problem, this was about 6 or 7 years ago, people were mechanically trying to pick out these features from the reconstructed density. These were structural biologists. And so one of the big advantages was any level of automation, or even semi-automation, that goes back and produces reliably good answers is a big acceleration to the structural modeling activity. So this technique uses a couple of different ideas. One idea was the fact that if I took a function and I, for example, a distance function from a shape, then I can actually reconstruct the shape from the distance function. Quite simply, I can take its level set. So if I took all distances from a shape S, then the distance where the function evaluates to 0 is exactly your shape S. But not only can I extract the shape, I can extract also secondary structural features or something called the medial axis from the shape. The medial axis is a set of points where you have more than one equidistant nearest point to the shape. So for example, think of a tube, and if I look at the axis of the tube, then clearly from the axis of the tube, the central axis, the nearest points are all the points on that tube. So hence the set of points, or the locus of points where you have more than one nearest point to the shape, is called the medial axis. And the medial axis can be reconstructed by a linear approximation and an intersection of two diagrams. One diagram is called the Delaunay triangulation, the other diagram is called the Voronoi diagram. So a Delaunay triangulation example is here, the Voronoi diagram is the dual. And these two, when I say the dual, it's actually like the orthogonal dual. Everywhere there's an edge in the triangulation, there's a bisector, which is an edge of the two-dimensional Voronoi diagram. When you intersect these two diagrams, you get what are called critical points. Critical points, and I'll come back to that algorithm that I skipped in the beginning, are exactly the points where the gradient of the function vanishes. So numerically, clearly what you could do is you can compute the function, you can approximate it by some spline basis, and then you can go back and compute its gradient and find its critical points by evaluating it numerically. But this gives you a piecewise linear and a combinatorially fast approach to computing the critical points. You take your shape, you compute the Delaunay triangulation of points sampled on the shape, you compute its Voronoi diagram, intersect them, you've got all of the critical points. Furthermore, from these critical points, you can actually classify them based on the number of directions where the function increases or decreases. So if I was to take a critical point and say, let me look in the neighborhood of the critical point and see how does a function behave at a critical point, then at a minimum, all directions are rising in a three-dimensional function. At a maxima, all directions are falling. And at a saddle point in 3 dimensions, you can have one of two cases. You can have two directions falling and one direction increasing, and the other case of two directions increasing and one direction decreasing. So this is what you can start to call the index of the critical point. And this index, in some sense, gives you the first order behavior around this critical point. And so this theory, also called Morse theory and then other versions of discrete approximations of Morse theory, is what one then utilizes to compute it. And so what one showed was that if I go back and compute the Voronoi and Delaunay computation, identify the medial axis, I go back and compute the critical points by the intersection, I can then classify these medial axes into what are called unstable manifolds coming out from index 1 saddles and unstable manifolds from index 2 saddles. And these are essentially axes of my helices, and two-dimensional manifolds are the beta sheets, the medial axis of the beta sheets. So does that solve the problem? It solves the problem only partially. The data is quite noisy. The input data, the reconstructed data also has noise. You solve it using a numerical optimization technique. And so given the noise, when you apply such linear approximation algorithms, you get close to the final solution. The good news is that you get a superset of the final solution. You get a lot more than what you need, but then what you have to do is apply filtering methods and other techniques that allow you to really segment out the actual helices and the actual sheets. And you have a lot of data that you validate this with respect to. You can take known structures, you can go back and compute by a simulator, a density map, and then you can go back and apply your algorithm and then check with respect to the known data. And that's what one tends to do. Another nice problem that still requires a more robust solution, and that's why I thought I'd mention it here. But you can not only extract what are called secondary structures, you can extract tertiary structures. Sometimes these proteins arrange themselves into what are called horseshoes, sometimes into circular motifs, and all of this can be computed by these stable and unstable manifolds. So this is what I was calling my group of challenges 1. How do I take microscopy data and from first principles try to come up with a 3-dimensional reconstruction and then using a whole bunch of computational geometry and image processing algorithms and perhaps some optimization to go back and detect and build structural models. But now that I have the models, what do I want to do with them? Well, one of the application arenas that we got interested in is to try to understand the problem of molecular recognition. How does one protein recognize another protein? And what are the forces that are guiding this recognition, or what are the energies that are guiding this recognition? So some of the computer science-like questions is what I'd like to dwell on next. So one of the key things is we have to understand the notion of the biophysics of interaction and then go back and compute these models of interaction and something called binding affinities that I'll touch upon. And a nice computational problem is that of coming up with, it's a geometric optimization problem, find a suitable docking. So I give you two proteins. Can you predict how they would come together in their stable minimum energy configuration? So these two problems are what I'll get into a little bit of detail. But I should mention, there's a whole bunch of problems that are still unsolved. Given a structure, how do I know what flexible forms this protein will take? Is still only partially known. Again, back to the fact that we don't have a video camera, so we cannot observe exactly what we have. But we can take several snapshots. And from the snapshots, can we go back and understand? But we must know not only from the snapshots, we must know what the structure is. We should also know what the biochemistry of the molecules are, and also the biophysical forces that guide them to change different conformations. There's, of course, the classic protein folding problem and the side chain packing problem, and also the problem of how these viruses with their multiple proteins and nucleic acids suddenly assemble together to form these superstructures like spherical capsids or helical forms with their nucleic acids inside them. It all happens by spontaneous assembly. If we can crack that code of spontaneous assembly by understanding the ways molecules recognize each other, then we can go back and build such nanostructures very easily because we know what the guiding principles are. And so this, of course, will allude to a whole bunch of nanomaterial synthesis down the road. So I'll start in the middle because I don't have time to start at the beginning. So using the Latin phrase in media res, what are the main highlights of what are the governing laws, for molecular interaction? So one thing, molecules show atomic granularity. They connect atoms together with something called covalent bonds of various types, and they form stable machines of defined geometry. Motion is random and diffusive. However, any two molecules in a micron-sized cell can meet each other. You can imagine this room and it to be packed with rice, and we are the molecules, and I need to meet the person across in the room. I can still make my way through this crowded room and still meet the other molecule. And when spontaneous assembly occurs, groups of molecules all get collect together to form these structures. So it's amazing. They are clearly agglomerative forces as well as forces that where crowding actually benefits the entire process. Because otherwise, random diffusive motion, you probably never reach your target if you're always going to be randomly and diffusing in various directions. Inertia is negligible because you are dominated by the viscosity of the watery solvent environment. Water is essential and is also present in the nano world. Gravity is negligible because the masses are so small. The masses are measured in Daltons and are negligible. So all of our macroscopic views of interaction, of assembly, of parts, as well as how does one thing fit with another, has to be rethought about. So here's an example that I stole from Mike Gilson, who's a good friend of mine in pharmacology, where he produces random diffusive motion of simulation of a little poison, and I'll show you a little bit more about this poison. This is fasciculin. It's binding to an important enzyme called acetylcholinesterase. It's one of the off switches in our neuromuscular junction. But amazingly, it binds to this, and it finds it. This binding occurs in a very tiny pocket-like region. But this small molecule is really guided by the electrostatic potential surrounding this. And so computing things like electrostatic potential becomes an important feature. So there are several models, and this is what biophysical modeling has spawned over the last maybe 3 decades on what are the forces governing atomistic interaction or molecular-molecular interaction. So I've just picked one model here, but then some of the techniques actually propagate to even other finer models or coarser models. So this model is a thermodynamic energy model. It says there's something called the Gibbs free energy of a molecule, and that energy consists of 3 terms. It consists of the intramolecular energy, it consists of interaction energy of the molecule with the solvent, and it has temperature and the entropy term inside it. So molecules are a collection of atoms. They have charges on them, and they form bonds. So a positive charge and a negative charge with a bond is like a little magnet. And if I take a collection of them, they are like an amorphous magnet. Water is oxygen with 2 Mickey Mouse hydrogen ears on it. And so water is like also a little magnet. It can quickly hydrogen bond with another molecule. The hydrogen positive can attach an ionic hydrogen bond with the oxygen. And so that's the magic of water. If you have water molecules in steam, all you have to do is lower the temperature, and they condense and they form a liquid where they have these hydrogen bonds. And if you lower the temperature, they form crystal. So hydrogen bonding occurs fast, requires very little energy, and it's kind of spontaneous. But it's what glues a lot of things together. And so the fact that it's also polarizable, because if you have a magnet and you have tiny little magnets floating around, then this larger magnet can cause a lot of water, tiny magnets to line up along the North Pole and those along the South Pole, the North Pole being positive charge, say, and the South Pole being negative charge. So how do you capture all of these physics of interactions? So one empirical model is, let me think of the charged molecule with its bonded structure as having the following energies. It's got bonded energies of bond stretching, bonds rotating, and so on. It's got electrostatic energies of there's a positive charge, you cannot get too close to another positive charge because it repels by Coulombic forces. It's got also called van der Waals-like energies, because atoms are really has a proton which is positively charged and is surrounded by a negative cloud of electrons orbiting around it. So a non-ion, but a single atom has then the potential of attracting the positive proton in one atom, attracts the electronic cloud of a neighboring atom. And vice versa, this proton attracts the electronic cloud. But they can't get too close. When they get too close, then the electronic clouds repel each other. So there's an optimal distance at which you got the correct balance of the positive attracting the negative, and then the negatives and the positives repelling each other. And that's captured empirically by this term. Now, if I just look at this from a computer scientist viewpoint, I say I have to estimate this term. This is clearly dependent on the configuration of the atoms. And so for every configuration, I have an energy. And as soon as the configuration changes, I have to recompute the energy. So I better make this computation very, very fast. Because typical molecules, proteins, are in the tens of thousands of atoms. And so if I have to compute these, but if I start to look at them, I say, oh, look at these two terms. They're both quadratic in nature. And if I have 10,000, quadratic means 10 to the 8th in my computation. And that's prohibitively large. And if I have to do this for every little change the molecule has to take. Hence, can I speed up this calculation? And so we put on our computer science hat and said, I can always speed up this calculation by doing what we call 1 plus epsilon approximations. If I allow for some inaccuracy in my summation, then I can do it in linear time rather than quadratic time, but I've incurred a bigger constant in front of it, and the constant is regulated by the approximation error. What's also more relevant, and I'll tell you a little bit more detail in a second about this, what's also more relevant is what about the interaction with the water and the polarization effect. So each of these terms is compute intensive, and I'll tell you a little bit more detail about the polarization. Finally, to work out the dynamics of the molecule, I not only need this energy, but I need the derivative of this energy. And if I need the derivative of the energy, this vector of force as a derivative of each atomic position, this force is what's going to guide the next configurational change. Hence, molecular dynamics is still obeying to do this accurately and fast and stably for various molecules. Today, the best of science, and I mean the collective computational sciences, can probably do a simulation of a average size protein to up to 1 microsecond of simulation. And 1 microsecond is not long enough to tell you what the total flexibility is. Some of the molecules move in the tens of seconds. So we are still a far ways to predicting flexibility of a molecule, just because our computational, with all the firepower that you have, you can only do about 1 microsecond. So this is again another huge challenge to go ahead. So let me, because of interest of time, I'll just briefly allude to the fact that the polarization term is actually a more challenging term to calculate. There are two kinds of theories. One is called the Poisson-Boltzmann theory. You have to solve this differential equation. But we are very good now in computational sciences to solve differential equations stably and fast, and we're getting better and better, and that's what I'm showing you here. And of course, it's building upon a lot of work by several other groups. There's also this calculation of generalized bone theory, where you're trying to compute this approximation to this polarization term. And this formula is a formula consisting of not only the distance between the atoms, which are n squared, but also calculating each of these radii called the bone radii. And each radii is an integral in its own right. So this calculation is order n to the fourth if done naively, where n is the number of atoms, or m to the fourth. But again, using clever algorithms, we can actually bring it down to m log m from n to the fourth. And this is necessary because we're going to use this always inside this loop. So just to give you a idea of how these algorithms are made, I'm going to skip this part on these algebraic splines, so they are dear to my heart, and tell you about one idea that is used and helps you develop these fast methods. That's this bone radius calculation. So think of this bone radius calculation. You have to compute this radii for each of the atoms that make up your molecule. And this radii is basically an integral or a summation from all points of a molecular surface. So the molecular surfaces that one can compute using various techniques, which I skipped, but let's keep that aside. Once you compute that molecular surface, you can do a sampling of the molecular surface, and these are what are called quadrature point sampling, that makes this approximation, this integral, be within a closed epsilon substituted by this summation. So this summation is a quadrature approximation of this integral. But the essential combinatorics is you have to compute the sum of all of these distances from every point on the surface to every atom center, and you can do this for all the atomic centers. So you have this n squared sums, linear sums to each atom, and you have to do this for every atom. So I'm putting that here. So here are my set of points on the molecular surface, and these are my atomic centers. So I developed two octrees. Octrees are simple subdivisions of space that capture such that you have a constant number of points in each cell. So you can selectively subdivide. This I'm showing you the quad tree in 2 dimensions. In 3 dimensions, it would be an octree because our molecules are in 3 dimensions. So I build an octree on the atomic centers, and I build an adaptive octree on the points on the molecular surface. And now I superimpose these two octrees and traverse them. and I come up with a certain classification of what we call near and far. So what I mean by near and far is this approximation idea that is not new, but it can be cleverly done, and it can be useful for lots of exercises, is the following. Every time I want to compute the distances from this set of points, in this example, to this set of points, I have some k-squared distances. The thing is, if these two cells are far enough apart, then I can probably take one distance, say the center of this box and the center of this box, and say all the k-squared distances are approximately the same as that one distance from the center of the two boxes. When will this be true? This will be true when these boxes are far separated. When they're very close, then clearly the distance between here and here is quite different from the distance between there and there, right? But if these boxes are far enough away, then yeah, all these points to all these points are roughly the distance between the two. So how do you go back and define far enough? Based on an epsilon that you're given. And how do you go back and subdivide? It allows you to then replace n squared calculations by only a linear number of calculations. So this entire technique allows you to calculate the entire summation in linear time. Linear in the number of points as well as in the number of centers of the circles. Sorry, I went backwards there for a second. So that's the essence of that algorithm, and that's essential, and I'll skip this part. But I can tell you about, this is what the computational chemist or the biophysicist likes. He likes to also see the implementation and says, oh, now I can really use this. And so we did this exercise for a 500,000 atom, cucumber mosaic virus shell. So we had to compute, of course, the bond radii as well as the polarization energy. But just to show you the timing difference, if I was to do things naively, then, and I measure the running time of our method versus the method, there's a very popular program out there which is called AMBER, which most computational chemists and all use. So we compared it to the naive as well as to AMBER. If I give you something like less than 2% error, then AMBER takes 1.5 hours to calculate, and the naive method takes 57 hours. While with 2% error, I can do the calculation in 9 minutes. So I get tremendous speed up by this algorithm, of course, going from n to the 4th to n. And I can now do this scalably. If I tolerate something like 15 to 20% error, I can bring my time down to 33 seconds. So this is the nature of then what we can call adaptive energy calculations. So when things are far enough away that I can come up with an approximation, I'll use a large error, and I can do the calculation, very, very quickly. And as I get closer and closer where I need to improve my accuracy, I can use the same algorithm to go back and calculate the energy effectively. So I skipped this thing about, because I'm looking at the time as well, about the potential calculation, which is the solution of this Poisson-Boltzmann equation. But going back to telling you a little bit about where these energies are becoming relevant, is when you're trying to understand, how molecules interact with each other, and in particularly these binding affinity calculations. So this is a molecule which is very similar to a neurotransmitter, an ion channel in our neuromuscular junction called the acetylcholine receptor. And this molecule is cobra toxin. And so cobra toxin binds to this and makes this molecule ineffective. And I'll tell you how it makes it ineffective. But here, all I'm showing you is this molecule, blue is positively charged and red is negatively charged. And so this electrostatic potential is like a gravitational pull. It pulls the toxin right into it. Of course, nature has evolved for predators and so on. And these molecules that have been developed in these venoms of snakes have the complementarity of attacking a weak link in various parts of our body, and hence they are toxic to us. Cobra toxins have this effect on the receptor, and another deadly poison is that of the green mamba, which attacks this enzyme called acetylcholinesterase. It uses a molecule called vesiculin. And let me show you a little bit about where this comes into play. So this is our neuromuscular junction. And this molecule is an acetylcholine receptor. So this is how the on switch and the off switch works in our body. Every time our muscle, we want to make any action, causes calcium channels to open up, which causes neurotransmitters called acetylcholine to dump into this cleft. These neurotransmitters bind to this molecule. It binds in little tiny pockets, causing this gate to open. This gate opening is a little pore region that allows for sodium and potassium ion exchange. And this ion exchange is important for electrical pulses to go down our muscle, just like electrical pulses going down our axons in our brain. The same mechanism is used. Sodium and potassium channel ion exchanges causes for pulsing, or spikes in our spike trains in our nervous system, also in our muscular system. So what cobra toxin does, it binds into the very region where the neurotransmitters would bind, and hence it makes this molecule from always being in the open state or in the closed state. It will never function after the poison binds to it. What is the off switch is, after a while, after this conduction occurs, this neurotransmitter gets popped out. And this neurotransmitter is cleaned up by an enzyme called acetylcholinesterase. This enzyme works at furious rates. It's just, it's in some sense diffusion limited. So its enzymatic activity cleans out this thing as a giant, vacuum pump. So what it does is it hydrolyzes acetylcholine into acetyl and choline, and that gets pumped back into our nervous system or into a neuronal cell, which causes for it to be conducted again after it's synthesized. So what the green mamba poison does, it blocks this enzyme from functioning. And it blocks it in the very region where the enzymatic activity or the active site of this molecule occurs. And again, it's caused by a giant attraction. The vesiculin has a positive potential, and this thing has a negative potential. And so one of the precursors is to then go back and see, what is the effectiveness of a poison, and secondly, can we come up with another molecule that can be like an antitoxin, and go back and get rid of this molecule faster than it can start to bind with this molecule. So the very nature of drug discovery is going back and searching for molecules that, once you identify a target, can bind to that target and bind to it more effectively than what its purposive nature is. And furthermore, to complicate things, just because we don't like things easy and we want to always solve more difficult problems, we also want to find a molecule that binds effectively to a target, but does not bind to all the good stuff out there. It has to weakly bind. So computation of the binding affinities, as well as the search, is then all coupled into what we call the docking problem. So what I mean by binding energy then is this relative difference of the free energies of the molecule. So if you look at, this is the free energy when the molecule is in its bound state. I'm just looking at A and B as two molecules. So if this difference is strongly negative, then these two molecules are better off being together than they are being when they are isolated. So this is a good measure of saying what is the affinity of one molecule with another. And what we have to do in the docking problem is, given two molecules initially, search for the best relative transformation and relative conformation that reveals a complex with the minimum binding energy. The minimum binding energy that I defined is this delta E, where E is all those terms that I showed you before. So we not only have to compute this fast, but accurately, and hence we also have to do this in the innermost loop of our search. So again, in the interest of time, I'll skip ahead, and I'm sorry I can't give you the details, but this entire exercise can be made into a fast correlation search. Correlations can be done very fast using fast Fourier transforms. You have to develop something called non-uniform fast Fourier transforms because molecules are atoms scattered in space, while most of the fast Fourier transforms were done for regular grids. But if you put all of that technology together, you can start to go back and show through various benchmarks as well as on real examples that you can actually come up with a fairly good model of binding affinity. And hence the docking problem is one way to go back and search for good leads that would perhaps then later on be synthesizable. Just to show you this chart, in our docking software we call F2dock, fast Fourier docking, and then the third version is fast Fourier flexible docking, that all of these energetic terms actually make a difference. So when we were comparing to a benchmark, we were seeing how good is our predictability. And just to give you a rough idea, the taller the predictability is, the taller the bar, the better your predictability. And you can see that the better predictability is starting to occur only when you add up all the terms. So once you've taken all of the biophysics of interaction, then you're always improving your predictability. But we haven't captured all the variables, and there are maybe several other terms that should be taken into account. So hence, accuracy of predictability is how accurate is our model of binding energy and binding affinity calculation. To close everything together, let me conclude by this project that we've been embarking on for the last 3 years only now, and that is we are trying to find a way to defeat HIV different to what had been done before. So HIV is a retrovirus, and this is just a schematic. It has an envelope, it has a nucleocapsid, it contains an RNA, it's a RNA-based virus. It attacks the T cell, which is the immune cell in our body. It has retrofitting machinery built in a setting. It has 3 important proteins in its nucleocapsid. There's something called reverse transcriptase. It retranslates the RNA into DNA. It has another protein called protease that goes and snips the DNA. And then it has another protein called integrase that integrates the snipped DNA into the genome of the T cell. The T cell is a very powerful part of our immune system. Our immune system is tremendous. Without that, we would not live that long. But what this virus does, it goes back and infects this T cell and then it lies dormant. The T cell's action, broadly, is every time there's an infection, there are markers that are developed and the T cell produces something called antibodies. These antibodies go back and bind to these antigens, and then there's a way of that molecule, that infection becoming harmless, and hence gets rid of it in some fashion or the other. But an infected T cell, instead of developing antibodies, develops multiple copies of the virus. So this is the life cycle that has been discerned by a whole group of very important people who have kind of understood this life cycle now. So then the first part of target discovery is to say, where's the weak link? So today on the market, there are 3 drugs that are used in a cocktail. One drug attacks the protease. So if you can knock out the protease, then you can make this life cycle, you interrupt the life cycle. If you knock out the integrase, or you knock out the reverse transcriptase. Now, these are all for post-infection. So what we've done is started to look together with Sriram Subramaniam, who's at the research arm at National Cancer Institute, is to see if we can go back and defeat the entry mechanism. And this entry mechanism is this little protein called GP120 that signals the T cell into fooling it to align its nucleocapsid to enter the cell. So what we've been looking at is designing a molecule and searching for good binding affinities of this molecule with this GP120, fooling it to thinking it's binding to the T cell, and instead it actually opens its envelope and dumps its nucleocapsid out into extracellular space. And the process, of course, is you have to use docking, you have to use these fast energetic computations to come up with good leads, and then you have to do validation of the entire process. And so my last set of challenges is about verification and validation, where you not only need to go back and calibrate every algorithm that you have done, which is what you call analysis verification, but you have to validate your models. When is your model good enough and when it's not? And the challenges are always model uncertainty, that you don't know whether you're capturing everything, and as you don't, you have to go back and come up with hierarchical methods of improving, and of course always data and computational noise. So with that I'll stop, and I think I've gone over by 1 minute, and I'll take questions, and I'll let you read some of the future directions that this area, I think it's rich enough for many more generations to come, and it's a great area where people like us, computational scientists, are very much needed, I think, to make an impact. So clearly in biomaterials, sensors, and other things. So thank you for your attention, and sorry to run over.
Thank you. So yeah, I mean, we are over time, so some people have to leave, but definitely would like to take some questions.
So in the first part of the talk, you're reconstructing these molecules based on data which seems very noisy at the beginning, and you're outputting something that looks like a very precise structure. So because of all the noise, there's really going to be some sort of uncertainty in the actual structure. Can you try and represent that?
Yes, we do. And in some sense, you know, when I talked about these mathematical terms, where everything is based on the configuration, it's really based on the geometry, the position of the structure, and the shape of the structure, and so on. But if I can quantify at the reconstruction what the error bounds are, and error bounds are like an error function, not just, you know, a uniform bound, then in each of my calculations, I can go back and tell you what is the, you know, emphasis I should take. And so some of the terms, and I didn't talk about that, have weights attached to them, so I'm not just taking a linear sum. And what we also tend to do is learn these parameters by looking at, you know, what weightage should we do with respect to the errors that we give. So the weights encapsulate the errors that we've made in our structure, so that all the functions, in some sense, don't, you know, are not unbounded estimations of the function, but bounded, error bounded.
You know, so you give this structure to some person, scientist working with it, you also give them the error representation as well?
So what we are giving them is, you know, what I was showing you, and I had to gloss over, we never give them a single answer. What you give them is the top 1,000 or the top 5,000. And these are called leads. And then there's another mechanism, and these leads have to be optimized. So they have to be optimized with respect to perhaps making a mutation, or adding another little residue or two to it in a compound-like structure. And then finally there's a synthetic chemist who must tell you, oh, I can actually synthesize this molecule. So the ideal team of people who take the leads is a computational person, a physical chemist, and a synthetic chemist who sit together and say, okay, well, this is possible. And that's the beginning of trials. Then you have to try it on animals and so on. But if you were to ask also the question, so where are we contributing? What we are contributing is in the initial lead generation. What is being done today predominantly is non-computational methods where every combination has to be tested in your lab before you can discover a lead. And that's searching for a needle in a haystack. And that's where we can accelerate.
So in your last slide, you briefly mentioned validation and replications. I'm just very curious on how you can validate your reconstruction of that molecule structures, especially as you mentioned, this inverse problem is very ill-posed, and perhaps you have non-unique solutions.
Good question. And so here are the techniques we use. So many structures have, there's alternate imaging methodologies to find them, like X-ray diffraction I mentioned. They give you the structure at perhaps better resolution, at atomistic resolution. So you take those models, you solve the inverse problem by first taking the known solution, taking the math, and comparing your answer with respect to those known solutions. And the better your predictability, the better your algorithm. So this is one way of which we can do algorithm verification. Now, the model that we use for the function, right, needs to be also validated. So there's a different process. So there the process is, if I'm predicting that this is the binding affinity, there's experimental methods and spectroscopic methods by which they can also calculate binding affinity. And we compare our computational binding affinities with respect to those. And hence we can come up with an error that our model of binding affinity is perhaps a little weak. And so we can then come up and try to come up with an improved model and validate that model. So both verification and validation, those are the techniques we use currently. Oops. Sorry. Yeah. Any other questions? Yes. Hi.
Excellent talk. Really interesting to see this kind of progress, especially given that there was a company 10 or 12 years ago called Physium Sciences. You may have heard of them. If you go to the website now, you find traces of them still around, but they have disappeared. And they were the first attempt to commercialize some of these ideas, do drug discovery through computation.
Right.
How close are we now? I mean, it looks like you're getting to that point, but
so
when
I
does
think
it become really commercially interesting?
This is, in the last few years, and this is it's like pieces have started to fall into place. Now, research has gone to the level that many of these empirical models and higher accuracy models are, have been validated, and people have discovered faster solutions to them. So the bane has always been, all of the docking software using very weak energy models of binding. Because the big problem is searching, and you're searching in very high dimensional spaces. Even for rigid objects, you're doing a 6-dimensional search. And if you allow for flexibility, which you have to, because you're given structures like this, but they come together like this, you have to have a flexible model. Then the energy computations, were very weak. But today, because these energy computations have been made better and better and faster using GPUs and so on, that it's becoming more commercial. So, Schrödinger is a company out in the East Coast that has gotten huge infusions of capital, and they bought out a lot of the smaller companies. That's one of the commercial packages. But a lot of the other work is in the public domain, just like ours, and is funded by NIH in resource centers. So all of the microscopy is in these microscopy resource centers that not only provide you data, you send in your samples, they also provide you some interpretation software. So that has grown, and so there's hope now. And because of that, even the big farmers are starting to create computational labs within themselves that would, they're paranoid always about their data and what problems they're working on, but they are welcome to take any possible software that can. So I think all of these forces are coming together, and I think as computational science is also coming together as a discipline, we'll have more bright young minds here attacking this. So I think we'll start to see an acceleration.
Okay, so let's, one more.
Yeah.
Okay.
As doing this calculation of energy, do you have any insight into how large an epsilon can be an epsilon approximation so you don't really change the underlying structure of the energy, so it's not doing something it's not supposed to do?
True. So, in the case of docking, it always is what problem you're trying to solve for what. So when I'm doing energy approximations for docking, I'm using the delta G. I'm taking the relative differences. And I'm using it to rank my candidates. So there I can get away with a little bit of error. Right? Because all I want to know is which guy is better than which other guy and which other guy.
So is 2% error okay?
Oh, sometimes even 10% error is okay. Because after all, the model is self-approximate, and so approximate, calculating the true energy, if you're within 10%, in a relative sense, it doesn't make much changes in your top solution. But when you're doing dynamics, you have to compute forces, which are derivatives on per atomic position. Then the forces should be error bounded. Hence you can go backwards and see how much error can you tolerate in the energy, and there may be less than 2% energy differences. Because you're taking derivatives, your energy calculations have to be very accurate. So it's always based on the quantity of interest that you're estimating, and then you have to go back and look at it. But if you have a scalable algorithm, then you can use it carefully.
Okay.
Thank
So
you.
let's thank you again.
Yeah. You like it, huh?
Yes.